that we penetrate the cabinets.
No vaccine, no job.
I think we set the standard because in Switzerland, for those who do not know, there are no restrictions anymore. So we set the standard that we require vaccination. Now, how many vaccinations do we need in the future?
It's a very good question. And I think, first of all, I think we will need vaccinations in the future, but also I'm concerned that the compliance of the population with the recommendations of the experts will not be very high. More people did the first dose, and then lesser did the second, and then lesser will do the third and the fourth.
An effective defense. They don't seem to work against me.
Sadly, if you refuse to get the fourth shot, or if you've refused to get the third shot, but you complied with shots 1 and 2, you're now unvaccinated.
I've gotten, I think, 2 or 3 boosters now after the initial vaccine.
Yeah.
It lasts a while. Are we going to have to get it every year, or are you developing something that might actually last longer?
Yeah, it doesn't last the current ones, and also doesn't last the disease. For example, if you get disease, you can get disease again after 3 months with the same even variant, not even absent a change of the variant. That's why it's very difficult for this virus to eradicate. I don't think it will. But we are working to see if we can get an annual vaccine. The number one priority is to have a vaccine that lasts a year. So once people know that it is once a year, I do it every autumn, for example. An annual vaccine will be ideal because people will remember, they can do it with other vaccinations like flu, and can last.
And I think there are also certain attempts to combine it with a anti-flu vaccine.
That is another very good way to make it, to improve compliance. If someone thinks that I'm going to go to the doctor or to the pharmacy, but at least with 1 shot I will get 2, flu and that also.
A flu, you raised it.
Yeah.
Where are you in developing? Because I think that's what you were originally doing with BioNTech. You were developing mRNA for flu. Where are you on a flu vaccine based on mRNA?
Oh, the studies are running. They have completely recruited. We are waiting for cases as they accumulate. Means that people have been vaccinated, placebo, vaccine, and then the disease, some of them will get disease. And then we are waiting to unblind the data to see what is coming. I think will come in this year, in 23.
Well, that's what I was going to ask.
You're a drug dealer.
No, what? How? What?
Now, I come back to a technical question. When you produce those very complicated medicines,
Cocaine?
those very complicated medicines, but mRNA,
Methamphetamine.
but mRNA, some people say the knowledge is actually not the intellectual property of the product, it's the production itself.
You get the maestro cooking again, I'll make it worth your while.
Knowledge is actually not the intellectual property of the product, it's the production itself. Could you say something about it?
I think there are across the board. For example, we don't own right now for this vaccine intellectual property. It is BioNTech's, right? But we developed the manufacturing process to be able to do it.
But I'm a manufacturer. I'm not a dealer.
Oh.
Per se. It doesn't mean.
We developed the manufacturing process to be able to do it. So all of that are things that they count. In general, I think that the mRNA technology, it is a very powerful technology.
So I'd like to thank you all for coming today. I'm going to tell you about a technology that is revolutionizing biology and offers the potential to cure human genetic disease. So this story really starts with DNA.
Jennifer Doudna and other scientists discovered a new way to edit DNA.
In general, I think that the mRNA technology, it is a very powerful technology. I don't think it's a panacea. I don't think it's the holy grail.
Editing DNA with precision has been a holy grail for scientists for decades. The DNA, of course, encodes all of the biological functions.
You probably know that DNA is the code of life that gives cells the ability to function, gives organisms the ability to develop, But when there are mutations in that code, it can also give rise to disease.
I don't think it's the holy grail. I don't think that will treat all diseases in the world.
And so some of the mistakes in DNA cause disease, genetic disease. If we could go in and fix those mistakes, we could save many, many lives and get rid of these diseases.
I don't think that will treat all diseases in the world, but I do think that we have just scratched the surface of what we can see.
Using CRISPR to help cure disease is not very controversial, but using CRISPR, you could actually change the DNA that would determine your baby's eye color or skin tone. Most scientists agree that this is something that we should not do. You know, nobody would have predicted that I and Dr. Fauci would be so prominent
about whether or not people were getting infected with bad viruses. So in a very minor collaboration, as part of a subcontract of a grant, we had a collaboration with some Chinese scientists.
Chinese scientists showed recently that they could even use the CRISPR technology to change genes in human embryos. And scientists in Philadelphia showed they could use CRISPR to remove the DNA of an integrated HIV virus from infected human cells. The opportunity to do this kind of genome editing also raises various ethical issues that we have to consider, because this technology can be employed not only in adult cells, but also in the embryos of organisms, including our own species.
One of the features of this fourth industrial revolution is that it doesn't change what we are doing, but it changes us.
The first applications of the CRISPR technology are going to happen in the blood, where it's relatively easier to deliver this tool into cells compared to solid tissues.
It changes you if you take a genetic editing.
We downloaded the sequences from the Chinese government, we put it into our proprietary algorithm that generates the vaccines, and in 2 hours we had the vaccine already designed and it went straight into manufacture.
With the incredibly fast development of the vaccine, could you tell me the story from the engineering to the science to the human story of how you could do it so fast? By November, you even had the ambition to do it by October. It was in the initial days. How do you,
8 days later,
in that time, how do you show that the vaccine is safe and effective, given that I think previous vaccines have taken years to do that?
Yeah. The vaccines take years to do that.
how much is the mRNA technology protected by patents or...
I think it's the holy grail.
How far are we away from one vaccine that's both COVID and flu together?
First, we need to have a flu. And if we have a flu, already we started experiments.
And the remarkable thing about this experiment is that when these mice grow up, you can test them and show that every cell in the body has this single genetic change that gives rise to the white coat color, but otherwise they're absolutely normal. So they are normal mice, no mutations elsewhere in the genome. This is a type of experiment that used to take at least a year to create a mouse like this, and now it can be done in a few weeks and by people that don't have to have special expertise. So this is a technology it's been very exciting over the last 2.5 years to see this technology taking off.
The technology will be used for gene editing, to be able, if people are born, they have a genetic disease, that is right now, it is untreatable, and in many cases a death sentence for those. There's nothing that they can do about it. Now we have some hopes that maybe that also can work. So we will see a lot of advancements, exactly because so many people are working. We are working on that as well, on all of that, but there are also many others, so that increases the chances that something will come out.
The technology is being used to make very precise changes that allow us to study the way that these changes in the cell's DNA affect either a tissue or, in this case, an entire organism. Now, in this example, the CRISPR technology was used to disrupt a gene by making a tiny change in the DNA in a gene that is responsible for the black coat color of these mice.
Laboratory mice, the genes have been spliced.
Imagine that these white mice differ from their pigmented litter mates by just a tiny change at one gene in the entire genome, and they're otherwise completely normal. And when we sequence the DNA from these animals, we find that the change in the DNA has occurred at exactly the place where we induced it using the CRISPR technology.
If we could go in and fix those mistakes, we could save many, many lives and get rid of these diseases.
This is no longer science fiction. Genome-engineered animals and plants are happening right now. And this puts in front of all of us a huge responsibility to consider carefully both the unintended consequences as well as the intended impacts of a scientific breakthrough. Thank you.
So there's no risk at all of causing the disease. One final way that's new and is promising is called the RNA vaccine. With RNA and DNA, instead of putting that shape in, you put instructions in the code to make that shape.
Nobody will be safe if not everybody is vaccinated.
So this is urgent, and it's going to require incredible collaboration. It's going to have to go to 7 billion people.
One issue that really grabbed me as urgent were issues related to population, reproductive health.
But did you come to reproductive issues as an intellectual?
When I was growing up, my parents were always involved in various volunteer things. My dad was head of Planned Parenthood, and it was very controversial to be involved with that.